Using this protocol to remove the intersample variation can offer insights into the differential regulation of macromolecules based on the intrasample discrepancies between mRNA and protein levels. Using the same sample to isolate DNA, RNA, and protein is an attempt to reduce variability, improve reproducibility, and facilitate interpretation. Benefits include saved time and resources at time of sample collection and cross-sectional analysis of valuable and limited samples.
To begin, seed 1,000 worm eggs in one 10-centimeter plate with appropriate growth conditions. Incubate at 20 degrees Celsius for 72 hours. Then, wash the plate with 5 milliliters of M9 buffer and transfer 1,000 adult worms into a tube. Centrifuge the worms at 1000 G for 1 minute. Discard the supernatant and transfer the pelleted worms with 1 milliliter of M9 buffer to a 1.5 milliliter microcentrifuge tube. Centrifuge again at 845 G for 1 minute, and discard most of the supernatant. Store the pelleted worms at -80 degrees Celsius for more than 4 hours. Next, remove any supernatant from the thawed pellet. Add 1 milliliter of cold GTCP reagent, mix well by pipetting up and down, and place the sample on ice for 10 minutes. Then, add 200 microliters of cold chloroform, shake the tube vigorously for 15 seconds, and leave it at room temperature for 3 minutes.